COVID-19 since this spring and almost choked. And it's really feel like we are kind of united by attending this kind of meeting. Okay, the time is real short. So actually, I have prepared two topics for today's talk, and the first one is the Bayesian analysis of the breast cancers. And actually, I have prepared the second talk as an integration of the single-cell RNA sequencing with the clinical sequencing of adult T-cell leukemia. But I may have to skip the second and other unnecessary slides because of the time. They have announced that my talk should be finished within 15 minutes instead of 25 minutes, which I misunderstood, and so I have to squeeze. Anyway, so okay, let me skip some unnecessary introductions, starting from my personal introduction. I'm in charge of the sequencing co-lab at the University of Tokyo in close collaboration with the National Cancer Center in Japan. And some references are here, and the latest one is edited by Uyam, and which is really an exciting experience to me. And perhaps some of you may are interested, quite used to looking at these slides. General introduction of the importance of to analyze the microheterogeneity of cancer tissues by single cell analysis and spatial transcriptome analysis. And let me skip it. And some general introduction for the Bayesian spatial transcriptome analysis as a kind of the latest version of the single cell analysis. Let me skip it. And some unnecessary introduction for the spatial analysis of the normal cells. Okay, let me start from here, which is the first topic of mine. And this is our recent attempts to elucidate heterogeneous transcriptomic feature of the breast cancers. Of actually the ductal, precisely speaking, ductal carcinoma in situ of the breast. That is the early stage of the breast cancer. This may be the introduction, which I can skip, because I guess some of you, I assume that some of you may not are so familiar with the ductal carcinoma in situ of the breast. And the ductal carcinoma in situ, or DCIS, is kind of the early stage of cancer from the normal ductal lumen to the invasive carcinoma. Ductal carcinoma in situ, DCIS, is here, which is just in between the apical hyperplasia and before advancing to the invasive carcinoma, and still in non-invasive type of the carcinoma. And there is a chance that the DCIS may potentially develop into invasive ductal carcinoma. So currently the first choice is the surgical operation. And maybe its target for the preemptive surgery. But the concern is that the substantial number of DCIS cases may never progress to IDC. So the current preemptive surgery might be a potentially over treatment. So at the first place, we wanted to know the mechanism, how some of the DCIS may develop to invasive carcinoma, others not, and to further understand the mechanism of the initial events, which drives the benign tumor to the more advanced tumors. And because those initial events may take place in a quite limited area of the cancer. So we made use of the spatial transcriptomic analysis using Bayesian this time. So, okay, here. So this is the mutation spectrum of DCIS. We have analyzed so far 74 cases, and the mutation spectrum showed up like this. And some cases relapse, even though this DCIS is generally the non-invasive, and mostly the patients' outcomes are relatively benign. And the mutation spectrum showed that all the relapsed cases had the GATA3 mutations in GATA3 gene. And on the other hand, non-relapsed cases are more associated with the mutation in PIK3CA. So please remember that the buffs of GATA3 and PIK3CA both in DCIS were around 5%. So those mutations took place only in a limited number of cancer cells. So we wanted to know what's the difference between the GATA3 mutation cases and the PIK3CA mutation cases, and conducted the Bayesian analysis. And we firstly selected the case B from the this is the case B, and the initial question was why this patient developed relapse, experienced relapse, even though she doesn't have any GATA3 mutation, genomic mutation. So this is the patient background, and 56 years old, and the ER positive rates were 90%, which was confirmed by the immunohistochemistry at the protein level. And the stats of the Bayesian showed up like this, and because that case was the first case, which we applied the Bayesian analysis, and this number might have been overkill, but we generated 10 billion Ds. And the number of spots on the tissue was 2.5K, and the other stats appeared as normal. And it's a really well-designed system, and the Bayesian results showed up like this. And actually, what we did was just to follow the instructions of the space rangers and the other softwares and the instructions provided by the 10x Genomics. And initial clustering analysis, this is his HE staining, and the initial clustering was conducted at the K being 2, and blue parts were roughly separate the cells, transcriptome patterns, and the cluster at the clustering level of 2, and the blue dots were mostly from the cancer cells, and the orange dots are mostly from normal cells. Which was consistent with the HE staining, pathologically speaking. And when we further, separated the clusters into the finer level, and there are several subclusters emerged. I forget to mention. This is the gene expression levels for particular genes, representative genes, starting from estrogen receptor gene. And some parts show the intensive gene expression. And by the way, these parts, this area is under it's a kind of the more advanced part, IDC part. And historically, pathologically speaking, these are the DCIs, pure DCIs part. And cancer developed from here to within the milk duct to relatively invasive part, like the cancer spread out from the duct and spread into the certain area of the tissue, and finally developed to the invasive carcinoma. And the ESR and the epicam and the keratin and the other familiar markers, and LB2, which is a drug target of the breast cancer. And okay. And when we looked at the cluster level K equals 7, and we were able to separate the cancer cells into several subgroups, which was consistent with the pathological diagnosis of the cancer. And that is the DCRS, the benign part appeared, formed a cluster here, and the IDC, relatively the benign part of IDC showed up here, and the most malignant part appeared like this. And also, the several normal cells, stroma cells, were also clusters like this way, and we were aware that the normal cells were also clustered, depending on their microenvironments. And some these pink parts, pink dots, are from the surrounding region of the relatively benign, the DCI is benign, also benign part of the cancer cells, and these parts are from the just becoming malignant part of the cancer, and those are from the invasive cancers. So the information was far richer than the TSNE because these are no more than it's a TSNE more or less. So first we wanted to analyze what's the difference between those three clusters, the DCIS and malignant and the IDC, the early stage IDC and the late stage IDC, and we conducted the clustering analysis, and the following results are relatively robust regardless of the clustering methods. And actually, the clustering results showed up like this, and these are the relatively benign part. These are invasive parts, and these are more or less, pathologically speaking, those parts are DCIS. And the fine clustering analysis showed that these parts can be separated into three clusters. And we were interested to see that particularly this part, because particularly this parts is pathologically within same duct and the same region at least, but transcriptomically, from the viewpoint of the transcriptome features, they belong to two separate clusters, and the surrounding region, surrounding dots are separate, classified into the cluster 1, while the inside was separated into, categorized into cluster 2. So when we look back to the histopathological image, and we were noticed that the inside was really like the benign-looking cancer in terms of the karyotypes and the shape of the cells, and the surrounding regions are just becoming malignant and started to invading to the surrounding area. So once we collected this clustering information, the following was the relatively straightforward gene enrichment analysis, and we found that the cluster 1, which was the invasive part, just invasive part, showed the transcriptome signature of the GATA3, and actually the GATA3 expression was repressed compared to the cluster 2, which was in the central part of this milk duct, and which was represented by the differential gene expression analysis. And we further conducted the gene enrichment and pathway enrichment analysis, and found that the cluster 1 showed a significant enrichment of the epidermal mesenchymal transition related genes, angiogenesis related genes, and estrogen response genes, which were all the genes related to the malignancy of the cancer cells, and shown like this. And this is because so we suspected that because of the GATA3 transcriptome repletion, so it triggers the malignant transition of the DCIS to the IDC as an initial event. So the next case, in the case B, the change took place at the transcriptome level. And because we selected that case because the BISIM was designed to do the transcriptome analysis. And the next case was we focused on the case A, which had a genomic mutation in GATA3. And we conducted a similar analysis and found that these are clustering, the results of the clustering analysis clustering analysis. So in this case, we were kind of lucky because the GATA3 mutation, that was a frame shift mutation, was located relatively around the 3' end of the messenger RNA expressed region. so that we were able to map the mutation on the messenger RNA tags. Yeah, mutation was represented on the messenger RNA tags. And so we were able to identify at which version the mutation was located by looking at the messenger RNA tags of the BISIM. The rest of the story was similar. So these parts show the GATA3 signature and which triggered the EMT, the expression of the enhanced expression of the EMT angiogenesis and estrogen responses, and the similar things was taking place while the case A was driven by the transcriptomic change, but the case B was triggered by the genomic mutation of the GATA3. By the way, we also found that the GATA3 mutation was relatively well associated with PCR expression, protein expression levels. So PCR may be easier to be utilized as a biomarker instead of looking at the GATA3 genomic mutation or transcriptomic changes in a clinical setting. So case C was the case where the PIK3CA mutation was found instead of GATA3 mutation. And in this case, the patient experienced no relapse, so the cancer was benign. When we analyzed this section by BISIM, we found that this was a relatively monotonous cancer in terms of the transcriptomic features and also the single cell genomic features observed by the single cell CNV analysis, and there's no enriched malignant looking pathways, and the geoterms were enriched. And the network analysis showed that the disrupted networks were at the minimum level, 2 networks were only disrupted. So it seems even also at the molecular level, this cancer looks relatively benign. So our current hypothesis is that the genomic, we analyze DCIS, and we are thinking that the changes of GATA3 signaling is the key, which drives the DCIS to IDC. And mostly that change takes place at the mutation genomic level, but sometimes in some exceptional cases, the down regression of the GATA3 expression means the same thing with the GATA3 mutation at the genomic level. So we are still working on these slides because the information is really rich, and the analysis of the stroma cells surrounding normal cells are still underway. So there are significant difference between the surrounding cells and the malignant parts and the the invasive part and the more invasive parts, the most invasive parts. So there are several significant genes enriched for these regions. To summarize the first part and to report the DCIS project, we have conducted the transcript analysis as a first case of the BISIM application, and the spatial transcriptomic features of pre-invading and invading DCIS shows that the GATA3 mutation, malfunction of GATA3 mutation seems to be the key of the malignant transformation of the DCIS to IDC, either at the genomic level or transcriptome level. So please refer to this paper, which we recently submitted to medRxiv, and under the Nagasawa did most of the work for this topic. So how many minutes is remaining?
Dr. Suzuki, actually we already passed about 20
minutes.
Okay,
passed. So I have to stop my talk here, and okay, that's it. Thank you for the kind attention.
Okay, thank you very much, Dr. Suzuki.
I'm sorry for the time.
have 5 minutes to discuss about your topics. So before we get the question from the audience, maybe I can start the question first, and maybe the limitation for your BISIM study BA, so resolution, right?
Yes.
So contain a lot of the cells together. But you showed briefly about the difference between regions. But do you feel that you need any further analysis for that to get the data for with the higher resolution?
Yeah, definitely you are right. That current resolution of this platform is quite limited, not perfect in other words. So we are kind of lucky because these spots the dots spots were just overlapping between the border of the malignant and the benign cancer cells. So like this. So the surrounding region. But so if the resolutions were finer, we may have been able to, it may have been more, we may have been able to analyze in more details which cell is which. I kind of, just the malignant transformation at the individual cell level, which is beyond the limit of, which is which is beyond the limit of the current Bayesian resolution. So the cancer analysis, I have skipped the introduction, but cancer is a kind of easy target because the cancer, cancer region is like a relatively big one, and the normal tissue is not like that easy. When in another story, we apply the Bayesian platform for the nephron, that analysis of the kidney, normal kidney. And nephron is like here, and the resolution is like here. So if we are lucky, we may find a spot which just overlaps with this nephron, but not always the case. So the resolution is a relatively an issue for this when we think about applying this Bayesian platform for the spatial transcriptome analysis, strictly speaking.
Okay, here's one question from the audience,
Yep.
from Kwan from Australia. Actually, he asked about is it possible to predict the expanded progression of cancer within the tissue using Bayesian data?
That's exactly what we are trying to right now. But the obvious, limitation is that once the tissue is harvested from the patient, we will never able to know the outcome of that cancer tissue. So we have to, develop bioinformatics computational mean to do that. So for that, we need more samples, a larger number of samples from a wider variety of tissues. So currently that project is ongoing, but I cannot answer that question right now.
Okay. Any other questions from?
I have a quick question. It's Ram. Hey, great talk. I was wondering actually, and maybe you didn't have the time to talk about it. Did you use this to also look at how the tumor stroma and the immune microenvironment may be changing at the beginning of DCIS, and how the EMTs that you see is, could you infer interactions between the tumors?
All those keywords are currently we are just looking at. And that's one thing I can add is that tissue I showed in my presentation were both relatively free from the cancer tumors, leukocyte invasions. So, and in other tissues, the fourth tissue actually, which I did not show in my presentation, or we particularly focus on the TILs, tumor infiltrating leukocytes. So the stories are quite different. The inflammation and the TILs and the cancers and the stromas and the players are more, and the environments are more complex.
Yeah.
So the simplest cases were the examples from which I showed.